Hello students, how are you doing? And now today our point of topic is enzymes. We will discuss about the enzymes and different related terms and characteristics of enzymes and how they act and the different inhibitors that inhibit the action of enzymes. So let's start. What are enzymes actually? Actually, enzymes are three-dimensional globular protein structure that are very important to regulate all the metabolism that taking place in any organism. So these are very important, and without them life is impossible. Enzymes are most important group of proteins which are biologically active. They tremendously increase the efficiency of biochemical reaction, and for specific type of reaction they are specific. It is very important feature of them that they are very specific in nature. For example, if one enzyme is acting to break one specific substance, then it doesn't act on other substance. Without these enzymes the reaction would proceed at very slow speed, making life impossible. That line is the importance of enzymes. Without these enzymes the reaction would proceed at very slow speed, making life impossible. And now we will discuss about the different terms like active site, substrate, activator, prosthetic group, coenzyme. So first of all we will discuss about what is active site. Actually, active site is nothing but the part of enzyme that actively take part in any catalytic activity. You can say that all the functioning that enzyme perform is limited to the active site. And what is substrate? The substrate is the substance that act on enzyme actually, and any substance that act on enzyme is called substrate, and after that it will convert into the product. So let's start with the definition. The catalytic activity is restricted to small portion of the structure that is called active site, and the reactant substrate is attached to active site consisting of only few amino acids. After that, very important thing, that is a cofactor and its function. What is cofactor actually? Actually, not all the enzyme in nature requires the non-protein part for their proper functioning, but some enzymes require a non-protein part for their proper functioning. That non-protein part is called cofactor, and it performs many functions. Like, it acts as a bridge between enzyme and the substrate, and it provides the chemical source of energy. And because cofactor is very important, without cofactor the enzyme does not work properly. So non-protein part called cofactor, which is essential for proper functioning of enzymes. The cofactors, now from this line there are functions of cofactors. The cofactor usually act as a bridge between enzyme and its substrate. Often it contribute directly to the chemical reactions which bring about catalysis. Sometimes the cofactor provide the source of chemical energy, helping to drive the reactions which would otherwise be difficult or impossible. So next we will discuss about the types of cofactor. There are actually three types discussed in our book. First is the activator, second is the prosthetic group, and third is the coenzyme. Actually, if this non-protein part is covalently bond, then it is called prosthetic group. If it is loosely attached to the enzyme, it is called coenzyme. If it is organic, it will be coenzyme, and if it is inorganic and loosely attached to enzyme, it is called the activator. So let's start with the definition of these two. The detachable cofactor is known as activator if it is inorganic ion, for example Mg positive 2, Fe positive 2, copper, zinc. If the non-protein part is covalently bonded, it is called the prosthetic group. If it is loosely attached to the protein part, it's called the coenzymes. Actually, the next is very important concept about the quantity that is required. Only small quantities are needed because, like enzymes, coenzymes also can be used again and again. Next is the types of the enzymes. and different names of the enzymes. The enzyme with cofactor is called actually whole enzyme. You can remember it by remaining the word whole. Actually, whole means complete, and complete consists of all the things. Like it contains two things, one is enzyme and one is non-protein part, and the apoenzyme is the only enzyme without non-protein part. So we can say apoenzyme is only protein part and holoenzyme is protein and non-protein part, that is cofactor. So the next is different sites of the enzyme. Enzyme important in photosynthesis are found in chloroplast. Enzyme involved in cellular respiration are found in mitochondria. Of course, where in which organelle there would be any action, there would be enzymes. Some of the enzymes which are involved in the synthesis of protein are an integral part of the ribosomes. So the next is characteristic of enzymes. Enzymes globular proteins, they increase the rate of reaction without being used. That is the reason actually they can be used in a small amount and can be used again and again. Their presence does not affect the nature or properties of the end product. Small amounts of enzyme can accelerate chemical reactions. They are very specific in nature. A single enzyme catalyzes only a single chemical reaction or a group of related reactions. They are sensitive to even a minor changes in a pH, temperature, or substrate concentration. Some enzymes require cofactor. Not all the enzymes in nature require a cofactor, but some enzymes require a cofactor for their proper functioning and lower the activation energy of the reaction. Actually, activation energy is the energy amount of energy that is required to start any chemical reaction is called the activation energy. So in this yellow box there is very important example of an enzyme. That enzymes are not produced in their active form. Actually, they are produced initially in their inactive form, and after or at the time of usage they are changed into their active form. Some enzymes are potentially damaging if they are manufactured in their active form. For example, pepsin is a powerful protein digesting enzyme and is quite capable of destroying cell internal structure, thus is producing inactive pepsinogen form of cell. It is converted in its active form only in the digestive tract where it is required to be active. So the next is mechanism. Actually, the whole comprise of one thing. When enzyme and substrate form, they form enzyme substrate complex, and after that enzyme and products are separated. Enzyme can be used again and again. This whole thing describe these things. After that there is very important the chain reaction. You can say it is very important short question. What is chain reaction? Actually, the chain reaction is that reaction in which a complex mechanism is performed with a sequence of events in a specific series. The successive enzymes containing these reactions are normally present together in a precise order of reaction such that substrate molecule can be literally handed on one from one to another enzyme. So it is actually in the one sequence. So you can say A to B, B to C, C to D, and D to E, and it will be called the precursor activation. And after that, if E can any enzyme that act on E and form the A, so you can say it is called the feedback inhibition. If the product or anything can act as an enzyme to start the reaction again, it is called feedback inhibition. So it is the mechanism. Actually, there is nothing more than that. So there are two types or two regions of active site. Active site can be divided into two regions. One is a binding site and another is a catalytic site. Actually, the name represents binding site actually recognize and bind to the substrate to the enzymes, and activated or catalytic site is the that site which transform the substrate into products. So we can read the active site of enzyme is made up of two definite regions, binding site and catalytic site. The binding site help in recognition and binding of proper substrate to the produce an ES complex, enzyme substrate complex, and activated catalytic sites the transformation of substrate into products. Next is very important two models about the working of enzymes. That first is the lock and key model and second is the induced fit model. Actually, lock and key model, as the name represents, it describes the as a specific key can be used to open a specific lock. In the same manner, a specific enzyme can transform only one substrate into the products. According to this model, it was presented by M.L. Fischer 1890, and as it states that as one specific key can openly a specific lock, in the same manner or specific enzyme can transform only one substrate into products. According to lock and key model, the active site is a rigid structure. It is very important MCQ. There is no modification or flexibility in the active site before or during and after the reaction. It is used only as a template. Later study did not support this model in all the reactions. And the next model is very important, that was given in 1959 by Koshland, and it is also known as a modified form of induced fit model, and it is also named as hand and glove model. Actually, he says that the active site can transform itself to that form that fits the substrate. He argued that when a substrate combines with enzyme, it induces changes in the enzyme structure. The change in structure enables the enzyme to perform its catalytic activity. You can say that actually on the induced fit model says that the active site can change itself according to the shape of the substrate and is flexible. Factors affecting the rate of enzyme action. Actually, there are 4 factors that affect the enzyme activity. First is the enzyme concentration. You should know, if one thing is constant, if substrate concentration is kept in excess or in constant, then you should describe about the enzyme concentration. If substrate concentration is in excess or in constant, then enzyme concentration is directly proportional to the enzyme activity or rate of reaction. So if we increase the enzyme concentration twice, the reaction will increase 2 times. So the rate of reaction depends directly on the amount of enzyme present at a specific time at unlimited substrate concentration. So this is all about the enzyme concentration. The next is substrate concentration. In the substrate concentration, there is actually one enzyme concentration is kept in excess and at a constant, and if we increase the substrate concentration, then of course the rate of reaction will be increased. You can see here the reaction is directly proportional, and it shows many active sites are present, and we increasing the substrate concentration, then reaction is increasing. At a certain point there is no increase, and after that the graph will become constant. Temperature. As we know, temperature is very important in any chemical reaction. So for enzymes, a specific temperature can increase the rate of reaction. So every enzyme has a optimum temperature. Optimum temperature means the temperature at which it works maximum. So actually what happens when we heat any chemical reaction, so it means we are actually giving activation energy, and after that the kinetic energy, and they collide themselves, and after that they increase the rate of reaction and vibration become more violent, and after a specific limitation the enzyme can be denatured. You can see here that if you increase the 10 degree Celsius temperature, the rate of reaction will double, and after a certain limit the reaction will be stopped and enzyme will be denatured after a certain temperature. pH value. pH actually very important. Every enzyme function on a specific pH. There are 2 types of pH change. One is a slight and one is extreme. In slight change in pH can change the ionization of amino acids at active site or ionization of the substrate. Actually 2 things are changed in slight change in pH. One is ionization of amino acid and the other is ionization of substrate, and at the extreme changes in pH cause the bonds in the enzyme to break and enzyme physically denatured. This is the table. You can remember it by saying the pose SSCPA. Pose SSCPA. P stands for pepsin, S stands for sucrase, E stands for enterokinase, S salivary amylase, catalase, chymotrypsin, pancreatic lipase, and arginase. So next topic is the last topic, that is inhibitor. So inhibitor, as the name represents, inhibit. Actually these are the things that inhibit the chemical activity of any reaction or enzyme. Irreversible, there are two types of the inhibitors. One is the reversible and one is irreversible. And reversible has also two types, competitive and non-competitive. Actually, irreversible inhibitor, they check the reaction rate by occupying the active sites or destroy the global structure. Actually, the name represent irreversible, so we can say that they occupy the active site by forming a covalent bond, or they physically block the active site. You can say if it is removed from the enzyme, then the new substrate, if attached to enzyme, it will not act. And after that we will discuss about the reversible inhibitors. they weak they form the weak linkage between the enzyme, actually, and their effects can be neutralized by adding the concentration of nutrients and different types of substrates. So the next is reverse types of reversible inhibitors. There are two types of reversible inhibitors. As the name represents, their effects can be neutralized. So one is competitive and one is non-competitive. Competitive means if they have competition with the substrate, then they will lose. You can remember it by competitive are those inhibitors which can lose if substrate concentration is increased at anywhere where the enzyme is also present and where the inhibitor is also present, then the substrate will bind instead of this type of inhibitor. And competitive, because of structural similarity with the substrate, they may be selected by binding site but are not able to activate the catalytic site. As we discussed, there are two sides of the active site, one is the binding site and one is the catalytic site. These attach to the binding site but they cannot activate the catalytic site and the product are not formed. The next is non-competitive inhibitors. Before we will discuss the non-competitive, the example of competitive is the enzyme is succinic dehydrogenase and the substrate is succinic acid, and the product it forms is the fumaric acid, and the enzyme is the same enzyme succinic dehydrogenase. And if instead of the substrate succinic acid inhibitor, competitive inhibitor is attached to it, then no product will be formed. And after that there is a last example of the reversible inhibitor, that is a non-competitive inhibitors. Actually, they form an enzyme inhibitor complex at the site other than the active site, and they physically retard the enzyme. And after that there will be the enzyme will be in that state, it will not act on any other substance or substrate. They form enzyme inhibitor complex at a point other than the active site. They alter the structure of enzyme in such a way that even if a genuine substrate binds the active site, catalysis fail to take place. Thank you. This was all about the third chapter of the enzymes. In the next lecture we will discuss about the new chapter. Thank you.